Good afternoon. My name is Cory, Pugh. I'm a sysadmin here at ClearEdge, and I'm going to talk to you about a company called Seleno and how they're using AI and machine learning to advance the production of stem cells. It's pretty cool stuff, hopefully. This next slide is a bunch of key terms that I'm not going to go over. I tried to in the dry run, did not work out. Basically, these are all the words that you want to go through and review in your own time. You can check them out, but I'm going to skip all these words and then use plain old words for the presentation. So, what is Seleno? Seleno is a startup biotech company, and they are focused on automating the process of generating personalized stem cells. See, I skipped that word, IPSC. But the goal is to use machine learning, laser physics, and stem cell biology to scale the production of stem cells. So stem cells are important because they help treat diseases that would normally could be terminal or degenerative. But the problem is right now the process is very labor intensive. It's a very expensive process. So Seleno aims to change all of that. They want to create a system where, using software-driven automation, they want to personalize stem cell production so that you have stem cells that are already unique to you. And this will be a cheaper process and would not require any immunosuppression because it's your cells. So how is this possible? So back in 2006, these two gentlemen here developed a process to induce mature cells back into stem cells. So basically, you can take adult cells and revert them back to the embryonic-like state, which then can be reprogrammed for other purposes. Which is important because before then, they had to use actual human embryonic cells, and that was unethical and controversial, so now they don't have to do that. So this is a huge step forward in stem cell research. So I'm going to talk to you about the current process in creating these stem cells, or gathering these stem cells. A scientist has to take a petri dish or a well plate of a cell sample, put it under a microscope, and on a screen, they have to visually inspect all of the cells and determine if they're high quality or low quality. So that, as you can imagine, this is a very labor-intensive process. And using a pipette tip, they have to scrape away all the low-quality cells. So imagine having to look at some cells and glue them with a pipette tip. Very inaccurate and not very precise. And it takes forever, so it's very, very expensive. So their process involves their own software and hardware that they generated. They take a well plate with a cell sample, put it into a machine, take a bunch of images, then they recreate a fluorescent image here, like at the bottom, is this black picture there. It recreates a fluorescent image, it tags every cell's location, and it determines if it's a high quality or a low quality cell. That goes to their laser that they created back in her Harvard studies. And that laser can target the individual cell and destroy it. So versus having to scrape away cells manually, this laser can pinpoint the individual low quality cell and destroy it. So much more accurate and faster. So this is a layout of the process. You take a label-free image, which is basically just an untagged cell sample. It generates a density map and a desired density map. So what it is and what it should be. The orange is all the bad cells, and then it creates a kill map, pretty much, kill or keep. So over here, everything in blue stays, everything in red has to go. And the laser can then pinpoint those individual bad cells and get rid of them. So next up, we'll have a picture side by side, before and after. So you can see on the before side, there's a lot more space. All that white area is all the low quality cells. All those get removed. On the right side, you have a nice red-white pattern of high quality cells. And the cool thing about this process is that every cell that gets processed throughout this is added to a database and becomes a data point. That data can then inform the machine learning algorithm and make it more effective. So it makes it faster over time as far as determining high quality versus low quality cells. And the thing that makes Seleno unique is that they are the only ones currently in the market who can conduct single cell processing. No one else has that technology. And that was something she discovered in her studies at Harvard. So some of the benefits of this is that one, it takes a tedious process and automates it, makes it faster, makes it cheaper. It produces an 80% yield of high quality cells versus the old process, which is a 10 to 20% yield. So much more cells, much faster, much cheaper. And the primary feature application of this is that there can be personalized stem cells for everybody. There can be cell banks that hold copies of your cells. And if you ever have a degenerative disease or a cancer and anything of that nature, they can then take your stem cells, reprogram them, and use them to help treat your own disease and cure a lot of diseases that are affecting us now. So this is something pretty cool because my aunt is dealing with the issue right now, and it'd be cool if she can have her own cells to help fix her eyesight. So 5 minutes.